and really excited to have Dr. Ishaan Gupta here with us, Assistant Professor from IIT Delhi, to talk about too many ways to die from COVID-19, results from the world largest cohort of lungs. So Dr. Ishaan Gupta works with the Functional Genomic Group at IIT Delhi and develops and deploy bench to code novel omics technology to measure biological phenomena with high resolution single cell, high precision isoforms, and high throughput technologies. So using this technology, he wants to understand the nature of biologically complex diseases. So with that, I would like to invite Dr. Professor Ishaan Gupta. So over to you, sir.
Yeah, hi, can everybody hear me? Hello?
Yes, yes, we can hear you fine.
Hi, is my screen visible?
Yes,
You can see my slides?
yes.
Okay, all right. Thanks to the organizers for hosting this talk. I think there is some issue with my computer, so I'm gonna basically be changing like this. I hope this is fine with everybody. Yeah, does
Yes,
this work?
yeah,
Because
it
at
works.
this point of time, I'm on
Yeah,
Linux
yeah.
right now, and yeah.
So can you do a full screen, or this will be the screen size?
Yeah, this is the screen size, unfortunately.
Yeah, yeah, that's fine. It's visible. I think it's kind of visible for the audience, and they can also, like, please let us know in the chat if you guys can see the presentation. Please let us know if there is an issue. It's visible. So yeah, it's visible. So so
All right.
you can go ahead, Professor
Yeah.
Ishan.
So thank you for having me. So today we are going to talk about I'm going to talk about some of the results in my lab, where we have looked at COVID-19 patients from India, and we have looked at their like lung biopsies and autopsies, actually autopsies after the patients are passing away from the disease. And typically there are three messages that we have that I can give immediately. Is that it's very important to actually do such studies on Indian patients, on Indian cohorts, and not only rely on the results that have been published from cohorts from outside India, because the genetics and how the patients, the environment that is the patients are exposed to is very different, which leads to very different kinds of results. So that's the first thing, first take-home message. The second take-home message that I can give, which is from the title itself, we find that there are two very distinct ways in which patients could be responding to the disease and therefore are succumbing to the disease. And the third thing is there is a big underappreciated role of the resident lung microbiome, or the microbes that are residing in your lung, that could also play an important role in COVID, as I will try to motivate in this talk today. So most of the work that I'm going to be presenting here was done in collaboration with Dr. Dipali Jain at AIMS, and was led by three students in my lab: Anshul on the left, uh, Anubhav in the middle, and Abhilash on the right. So this is a paper that was recently published in the journal DMM, Disease Models and Mechanism, about 2 months back. All right. So COVID-19 in India, there have been about 5 lakh people dead. So 5 lakhs would approximately be 500,000 dead, but there is no molecular data from India. So although we are a country with one of the highest disease burdens, of top 3 disease burdens, and a lot of, like, 500,000 people have died because of this disease. There is, I mean, very few data is there for exactly looking at the patient types or patient sub isotypes, looking at the molecular biology of the patient. There's a lot of clinical data, epidemiological data, but very little data from exactly how the patients look like under the microscope or at the molecular level. So this is what we are setting to do. This is what we had essentially doing. So we are essentially took 10 patients as controls, and 32 patients who had COVID-19, so patients who had died of the disease. And we took core samples from these autopsies very fast. So this is with a technique called MITS, is massively invasive tissue sample, massively invasive. I don't think it's invasive. Just that you have an injection which gets like you put an injection onto the lung samples or the tissue that you want to sample, and you can quickly it's minimally invasive tissue sampling. You can quickly get a tissue sample. So the idea was that we sequenced these samples and did the RNA sequencing. We did the quality control. We aligned these samples to the human genome. We found differential transcriptome analysis for these samples. So basically figuring out differences between the COVID samples and the normal samples. And besides the data from the human genome, we also had data from the microbiome that was resident in the lung, and that's what I'm going to talk about. So one major result from our study was that when we take a look at the gene expression level of the patients, we find two kinds of patients. So just to show you the figure, in this figure, you see that each dot is a patient, and the normal patients can be seen in the color red, and COVID patients can be seen in the color steel blue, right? And what you see is that when we are plotting the gene expression of these patients in higher dimensions, specifically capturing the first two principal components, basically characterizing how these patients could be different at the level of gene expression, we find that the patients are segregated into two parts, right? So on the left side and the right side of the graph, on the left you have sets of patients, COVID patients and normal patients clustering together, and on the right side you have only the COVID patients clustering together. So this suggests that there is a subgroup of COVID patients. So this is approximately 8 COVID patients who are actually very similar to normal patients at the level of gene expression. And on the other hand, there's the majority of the COVID patients, about 3/4 of these COVID patients look very, very distinct from normal patients. So what could be the reason for this is something that we try to figure out in this paper. So we had some clinical annotation for these patients, like whether they had some cardiovascular complications, whether they had diabetes, if they had liver complications or other comorbidities, and we did not find any signature. So basically these comorbidities could not be the reason why the patients were separated. So here we have colored, a no is colored in red and a yes is colored in this steel blue color, right? So what we find is that you cannot clearly see that, okay, one of the comorbidities like diabetes is more enriched in the patients which are similar to the normal patients versus patients which are extremely distinct. Although we do know that patients with cardiovascular complications and diabetes, from our own work, we have shown in Indian patients that patients who have diabetes are more susceptible to COVID-19. But that's at the clinical outcome level, but at least at the molecular level of how their lungs are looking, the gene expression of their lungs are looking, that's not the case. So then what do? We essentially go ahead and assume that these two groups of patients are distinct, and then we compare each of these groups with the normal sets of patients. So normal sets of patients here would be these patients in red. So what we do is we do a comparison of differential gene expression between this group, which is similar to normal, which we call G1, and this other group, which is very distinct from normal, that we call G2. When we do this, we find, as expected, lots of genes that are different between these two groups of patients. Specifically, there are about 1,000 genes in total that are different between these two groups. Some of them are going up, some of them are going down. And if you can see on this plot on the right, this is a typical heat map. On the first row, each column is a patient. The first row gives you the color of the type of the patient. So a light pink color means this belongs to the G2 group, or the group that was most distinct. And on the right side top, you have the dark maroon color, which would be patients which are in the G1 group, and you have this blue color, which patients who are normal, right? So if you look at the first, first rows, each row here is a gene. You see that there are certain genes which are only expressed highly in your normal patients, and they are not expressed in either G1 group of patients or G2 group of patients. Similarly, if you go down, you have certain genes which are only expressed in the G2 group of patients, mildly expressed in the G1 patients, and not expressed at all in the normal patients. And then you have another sets of genes which are expressed highly in both G1 and G2 patients, but are practically absent in the normal patients. So in order to understand what's going on, what separates these groups of patients and what unifies these groups of patients, we can actually take these lists of genes that we obtain after such a differential expression analysis and try to understand what are the common pathways that these genes belong to. All right, so when we do this, when we are basically separating these genes, differential gene expression, treating it separately, as expected for G1 patients, we have a much less number of genes as opposed to G2 patients. And what we find when we do pathway analysis is you have pathways such as fibronectin beta chain, serum amyloid, apolipoproteins are upregulated, these pathways, and genes which are relating to, say, scRNA or FABP4, they're downregulated here. And similarly, in the case of G2, some interleukins, some angiotensin enzyme, vitronectin, these enzymes are sort of upregulated, and a lot of SNO RNAs or specific genes are downregulated, right? There's a very little overlap between these two patients, just to confirm what we saw in the PCA plot. But in order to go deeper, we now try to look at which pathways are enriched for these genes. So specifically, I'm showing you here the pathway enrichment analysis for genes that are upregulated in G1 patients versus N patients. And what you see is that lipid metabolism is upregulated. Coagulation could be slightly downregulated, and there is upregulation of, coagulation could be slightly downregulated as negative coagulation, and neutrophil mediated immunity is slightly downregulated. And this is something that was confirmed in other papers, by other papers in smaller studies, by targeted analysis, and the papers are mentioned here. When we look for G2 patients, specifically one pathway which stands out is complement activation, pathways in xenobiotic metabolism, and peroxisomal protein transport. And this was also supported by some of the publications that came out early last year. Now, this essentially means that the disease that is being experienced by G1 patients and the disease that is being experienced by G2 patients may look very differently molecularly. So what could be the reason? Everybody would have heard of ACE2 receptor and TMPRSS2 protease playing a role in SARS-CoV-2. So we try to figure out if the expression of these two genes could be sort of indicative of what's going on. But for ACE2, definitely we did not see any major changes in gene expression. And for TMPRSS2 as well, there were certain changes, but when we correct for multiple testing, these changes do not hold that significant. There are other proteins which can substitute for TMPRSS2, which is a protease which helps the processing of the spike protein. When we tested these were also not really convincing and cannot distinguish between why these two patients are experiencing the disease differently. So the next thing that we did was we tried to enrich for specific gene sets that were known to be implicated in COVID-19, basically some pathways that are known to be sort of playing a role in the disease. Some of the pathways, you can see here, this was done by a tool called GSVA, which is basically gene set variation analysis. So as you can see, again, we have similar heat map plot, but instead of plotting genes as in rows, we are now plotting a score for a pathway in each rows. So if the pathway is upregulated, you would see a more green color. If the pathway is downregulated, you'll see a more purple color. And you can see there are pathways like unfolded protein response, which is known to play a role in viral infections. You see that this pathway is highly upregulated in the G2 sets of patients. So G2 patients are all these patients in green. Your normal patients are in blue here, and the G1 patients are in red. So you see in G2 patients, typically you have high unfolded protein response. There is high oxidative phosphorylation, fatty acid beta oxidation is also high, and all kinds of complement systems, classical, alternative, lectin-induced are all high. While in the case of both normal patients and G1 patients, these pathways are not high, right? Similarly, there are certain pathways which are high in the normal patients and in the G1 patients, such as these IL-1 cytokines, which seem to be low in the case of our G2 patients, the other patient subtype. So what this kind of analysis essentially tells us that there are certain mechanisms or certain pathways that are upregulated in both kinds of patients. So interleukin receptor, interferon response, JAK-STAT signaling is upregulated in both these patient types. But if you specifically look at G1, chemokine ligand signaling is specifically, such as the cytokines, is specifically upregulated in G1 patients. And here, if we go down to what kind of chemokine ligands, specific genes that could be found upregulated, we find that CCL19, which was shown to be a potential biomarker for patient stratification in the blood of COVID-19 patients, that was only upregulated in G1. While in the case of G2, you had very high presence of all the complement system, some chemokine receptors, TNF factors, TGF beta signaling, so on and so forth. So with this analysis, we can suggest that why is it that the patients look so different? Because the molecular biology of the lungs looks very different, specifically in the context of how chemokine signaling is working and in the context of the complement system in the G2 patients. And with CCL19, we actually have one way, one biomarker, which should be and which has been used for stratification of patients to tell which patient could be a G1 type or which patient could be a G2 type. Beyond this, we also can look at which specific cell types in the lung could be affected. So their lung is composed of many different kinds of cell types. And what we find is we can have gene sets with respect to each cell type. And what we find is that in the case of your COVID-19 patients, specific cell types like proliferating basal cells or alveolar epithelial type 2 cells, they seem to be disrupted. And if we make a sort of a plot for what kind of cells are changing their proportions between G2 and G1 patients, we specifically see that on this x equal to y line, your classical monocytes, lipofibroblasts, dendritic cells, they're all changing similarly. So either they're going down in both G1 and G2 patients, or they're going up in G1 and G2 patients. But two cell types, specifically proliferating basal cells and TREM2 dendritic cells, are positively changing in G2 patients, but they are negatively changing in G1 patients, which essentially means that suggests that these cells could potentially allow you to understand why these patients are behaving differently, and maybe even design distinct strategies to or drugs to treat these two patients differently. One thing that I started with in my talk was to sort of motivate that such studies have not been done in the Indian context and why they're important. So in this slide, I would like to motivate that. So there was a study in 2020 by Ninehold et al., where they took 22 patients, the biopsies from 22 patients, fixed samples. We have fresh frozen samples. And what they found was, they found that there are 68 highly upregulated genes and 30 significantly downregulated genes, because they were using actually a gene panel instead of doing RNA sequencing. And they found that specifically interferon genes were changing in this process. And what they did was they were able to subclassify their patients into these interferon stimulated gene high category and low category. And they said that there are two kinds of patients, the Caucasian patients that they were looking at. There are two kinds of patients in which have different kinds of COVID-19 manifesting. Now, when we do look at the same gene sets in our patients using GSVS scoring, we don't find the results to be replicating. We don't see won't find our patient two subtypes, the G1 and G2 subtypes, to represent the same subtypes that was found by Ninehold et al. So this essentially means that the way that the disease manifesting in the Indian patients is potentially different from the way it is manifesting in the Caucasian patients, or at least the we are using RNA sequencing. The difference in technology could actually be giving you different results. So therefore, this needs to be looked at. At least we are not able to reproduce the results that they have shown. So the take-home message for the talk so far is that clinical covariates have little impact on how the gene expression of COVID-19 is segregating between the two groups of patients. There are two potential kinds of these patients. The patients recapitulate the molecular aberrations or the changes in molecular biology which has been reported. So there is unfolded protein response, there is IL-1 inflammation, there is change in the complement pathway, there is changes in drug metabolism because most of these patients were under treatment. And there is downregulation of pathways like olfaction, which was one of the first signs of COVID-19, where smell was lost, and downregulation of dendritic cells and T cells, which are basically going to respond to the disease. And we found certain specific things in our COVID samples. There was enhanced lipofibroblast and basal cells and ciliary cells, and there were diminished dendritic cells, specifically IGF-21 positive alveolar type 2 cells and basophil mast cells. And there were certain cell types, as I said, which are distinguishing between our two patient types, and we can actually distinguish these patient types based on certain chemokine markers as well. The second thing that we try to actually look at is the remaining 20% of the reads which did not align to the human genome when we were doing our analysis. And these remaining 20% of the genes we found by aligning to custom databases of bacteria, viruses, and fungi, they're actually coming from the resident microbiome of the lungs. So what we essentially found was, if we compare COVID samples with normal samples, the microbiome diversity was massively downregulated. So not only did these patients have infection from the COVID virus, but as a result of infection from the COVID virus, there was also loss in the diversity of the bacteria that could be normally residing in the lungs. And you can also look at this loss of diversity in the context of a PCA plot, where the abundance of each genome that was detected, or each microorganism that was detected, is plotted. And you see that most of the COVID patients are on the right side, separating from all the normal patients, unlike at the level of gene expression. So there is definite dysbiosis in these patients, and that is something that one has to look at. So one of the things that we looked at in the case of dysbiosis was that we were able to actually detect complete genomes of the SARS-CoV-2 virus itself from the sample. So we were able to actually get 4 samples had high confidence detection of SARS-CoV-2 virus, and out of these 4 samples, in 3 samples, we were able to actually reconstruct the entire genome sequence of the SARS-CoV-2 virus. And when we looked at which lineage these viruses corresponded to, so they were into the Nextclade lineage 20A or Pango lineage B1.36. So this was the beta variant. So this was corresponding to the time of sampling of the variant. So this sampling happened in late 2020, early 2021. And it corresponded to the time where the samples were taken. So these samples were deposited. I'm sorry for the image quality. If I could have make it bigger, you would have seen it. So these samples were submitted on a GSAI, and we could see that all of them were actually sort of clustering with other samples that was submitted from India and from AIIMS, the hospital. And there were specific mutations that we could see, which would be interesting for people who are actually looking at how the virus is evolving. So as I said, the variant that we found were from this late 2020, early 2021 variant, which is during the time of sampling. So that sort of is a sanity check for the fact that we were actually capturing COVID samples of that time that we wanted to look at. Now, if we specifically look at how the dysbiosis is happening in these samples, instead of plotting all the COVID samples together, now I'm plotting the COVID samples separately. the G1 and G2 samples separately from the normal samples, and you see that both the G1 and G2 samples have has dysbiosis with a reduced Shannon diversity index. And although there is no differences at the level of the G1, G2 groups themselves, that the way that the dysbiosis has happened in the two patients is quite similar. So in both the patients, the bacterial composition or the microbial composition, actually we are also looking at viruses, is similarly diminished. And now if we plot the at the phylum level, how these in each individual samples, how these are looking at. So for example, each column here is normal samples. So the first 10 columns here are normal samples. The next 8 columns are G1 samples, and the next about 24 samples are the G2 samples, G2 patients. And what we see is that there is a similar kind of trend. There's nothing which is like outstandingly different between the G1 and G2 patients at the level of phylum. But now if we specifically go and take a look at the level of specific species that we were detected, there are some trends. So if you look at this heat map, again, each column is a patient. The patients who are corresponding to the blue column are normal patients. Patients who are corresponding to the red column are G1 patients. Patients that are corresponding to the green column are G2 patients. So now if we specifically look at these rows, which are marked as G1 versus GN, so these rows represent strains which are specifically found to be present in a high number in G1 patients and much lower number in G2 patients. So one of these strains is Staphylococcus epidermidis. So this basically is an opportunistic pathogen which is on our skin, and this pathogen was found to be more enriched in G1 patients than G2 patients. Similarly, there is Staphylococcus conii that was found to be more enriched in G2 patients. They were present in a slightly higher number, right? And the G2 patients also had pathogens like Aspergillus oryzae and some viruses. Similarly, there was other sort of resident microbiome which could possibly be protective, such as shown here. So all these bacteria, Mycococcus luteus, Ralstonia, all of these are the bacteria which are potentially depleted in the COVID patients. So what this sort of suggests is that there could be specific species of bacteria which can colonize the immunocompromised niche of the lungs of the patients who have been suffering from COVID-19. And interestingly, what you'll find is that the COVID-19 was only detected in like 6 patients. So the lungs had cleared out the infection. So basically, the take-home message for this part of the talk is that dysbiosis, you see dysbiosis in almost all the patients of COVID-19. And there is definitely enhanced presence of Staphylococcus infections in both the kinds of patients. And we were able to recover COVID-19 even in these patients during close to their, just before they gave up in 20% of the samples. And we were able to actually recover with high confidence in 4 samples. We were actually able to recover the entire virus itself. So the outlook that one could have from such an analysis is that working on a country-specific cohort like we have done revealed new subtypes. So it's important to actually go around, and whenever you are actually looking at some molecular phenotyping of a disease, it should be done across the world, across different populations, and definitely large population groups of the world should be represented, like India, which is typically not. What we find is that there are specific pathways which are changing between the patients, and there are specific suggestions that we have for stratification of the patients based on these pathways or based on the molecules that could be found in the blood that could act as a proxy for these pathways, such as CCL19. The fact that we have gene expression changes can be used to also do some in silico drug screening, something that I've not shown, but if you're interested, you can go and take a look at the paper. And also, we could find that these changes are specifically happening in certain cell types in the 2 different patient types, which can be used for designing targeted therapies. And then there is definitely a role of the microbiome in the lungs of the COVID-19 patients that needs to be further investigated. And we cannot rule out the possibility that the reason why the patients are not the patients had a tough time. Could be because of these infections rather than the COVID-19, because these patients mostly had cleared away the infection. We were not detecting COVID-19 in actually majority, 80% of the patients. So I'll end my talk here and I'll be happy to take questions. Thank you.
Thank you so much, Dr. Ishaan, for such an interesting talk. And I have some questions for you. We want to understand, is it the normal patients which were taken or healthy volunteers? Like normal patients suffering from disease, if yes, then what? Or normal patients who survive?
Yeah, so this is a good question. So we took basically autopsies of patients who were suffering from lung cancer. So typically when you were taking autopsies for, or not autopsies, or biopsies, so you would take some tissue which would be normal tissue and some tissue which would be cancer tissue. So we were using the tissue that was normal tissue from lung cancer patients. So these lungs were already inflamed. So they should have had some inflammation signatures. So those were the available samples that we had for ethical clearance.
All right, sir.